Good morning. Thank you so much for joining us. I'm Silvio Goodkin. I'm the Associate Director for Basic Science at the Moffitt Cancer Center in UC San Diego. I'm also the Chair of Pharmacology. I work here in the School of Medicine UCSD. And it's a real pleasure for me to have the opportunity on behalf of the QBI, Quantitative Bioscience Institute of UCSF, in the University of College Dublin, to serve as the host of Kimberly Luddy's lecture on team science in modern cancer immunology. The Kimberly's lecture is part of the existing partnership between QBI and the University College from Dublin, focused on advancing research on molecular networks on cancer and other diseases. So right now we are living in, and Kimberly will speak much more about this. We're living in what we call cancer immunology revolution. So now it's possible to turn on your own immune system to fight cancer. That's really unique. It didn't happen in history. And we are seeing for the first time complete and durable responses in many patients that would have never survived for a long time. Unfortunately, this doesn't happen to everybody. This is just, it's an issue. We know that sooner or later this is going to be fixed, but right now we don't know who would respond, who would not be responding, and we don't know from the few ones they respond and then they relapse. This is really what Kimberly will be talking about collectively in her unique expertise. So she first she received her PhD in immunology at Trinity College in Dublin, in Ireland, in focusing on ecology of cancer immunology. And she's currently works in the cancer biology and evolution program at Moffitt Cancer Center in Tampa, Florida. What makes her work unique is this ability to combine all of this information from immunologists, mathematicians, physicians, and being able to combine all this expertise and develop a systems biology type of approach to understand cancer response in those that don't respond, and how to overcome resistance to immune therapies. She applies evolution and ecological principles in cancer immunology. They are really unique. And so in that aspect, her ability to connect all of these different disciplines and the ability to bring this, in this case, even to the clinic, is really what makes Kimberly's work really unique. In that brief introduction, Kimberly, so thank you so much for joining us and looking forward to your talk. And by the way, don't wait until the last minute to write your questions. Don't wait for the last minute. Feel free to, in the Q&A, whenever something comes to your mind, just write it there. And at the very end, I will be hosting the Q&A with Kimberly. So thank you again.
Thank you very much for that introduction. It's nice to hear other people talk about how much teamwork goes into what we do. So today I will be discussing my talk, Team Science in Modern Cancer Immunology. Just get my screen working here. Okay, so we are talking about embracing complexity through collaboration. And as he said, this field really does require a network of interacting backgrounds. So if I can get my slide to advance. And so what it means is that we're looking at combining immunology, oncology, and evolution and ecological principles. So immuno-oncology is definitely at the forefront of cancer research for many reasons. One thing is the success in the clinic, and the other is that there's still a lot of room to grow. So there's a lot of gaps in our knowledge that we still need to fill to reach the level of patient intervention that we're looking to do. And in our department, we combine a lot of our cancer research with evolution and ecological principles. And hopefully through this talk, you'll kind of understand why. A lot of the funding and support behind the projects that we do are driven by Moffitt Cancer Center, which has a Center of Excellence in Evolutionary Therapy. The Physical Science and Oncology group at the NCI, which funds a lot of high-risk, high-reward team science projects where we work with physicists, mathematicians, computer scientists, clinicians, immunologists, ecologists. And also, of course, Trinity College Dublin, who supports a lot of the immunology and biochemistry work that goes into this. And the reason why we go with a team science and kind of a systems biology approach is that the cancer immune interaction is complex. As many of you know, there are lots of immune cells. They have lots of functions, lots of interactions between each other. They are interacting within the tumor and microenvironment, which consists of heterogeneous cancer cells, stromal cells, vasculature, lymphatics. So it's a really complex environment that requires reductionist immunology to understand the foundations, as well as high-level understanding of how these systems interact with each other. And a really great way to illustrate this is to show an interaction diagram. So this is just a subset of cancer cells interacting with stem cells and with parts of the immune system. And you can see just from one interaction diagram that the level of complexity that we're talking about is quite unique to these two interacting environments of the immune system and the tumor and its ecosystem. And building on top of that is that cancer immunology is also changing. As we get to know more about what the immune system does, how the tumor reacts to the immune responses, how our immunotherapies can affect these two systems, we really have started to build layers of complexity on top of this, what are helping our understanding. So it helps to really look at the history of immunology. Initially, the immune system was described as a way of identifying self versus non-self. So this is a pathogen kind of mutation-centric view of the immune response. So the immune system can recognize pathogen that's foreign and can recognize self so that it doesn't induce immunopathology and only starts to attack something that's considered non-self. And so this led to, for tumor immunologists, this led to what we call a mutation-centric view of the anti-tumor immune response. We know that tumor cells acquire a lot of mutations. We know that these mutations can stimulate an immune response. And so we look to exploit this with things like checkpoint inhibitors, which remove the brakes on an anti-tumor immune response, therapeutic vaccines, and antigen-targeted therapies, which drive the immune system to recognize malignant cells that are expressing mutated proteins that look unlike self. And building upon that is the danger hypothesis. This is Polly Metzinger's work from the 90s. Which really focused the tumor to start to look at a tissue-centric view, which is a wound that won't heal, kind of a viewpoint where we're talking about the microenvironment as a whole. We know from the danger hypothesis that the immune system can react even when you don't have the presence of non-self. So if you have a sterile wound that doesn't have a pathogen, you're still going to get tissue remodeling. And this really involves the innate immunity and can be attributed to adjuvants in therapeutic vaccines. So the danger hypothesis really builds onto the self, non-self. So you have stranger and danger, as we call it. So non-self pathogens or mutated cells, along with danger and disturbances that they're causing to the tissue, will alert the immune system. And modern immunology is really building on top of these two and adding another layer, which is considered kind of stress. So epithelial cells under regular homeostasis will not be under a stress response, but cancer cells have hallmarks. So we have things like genetic instability, high protein turnover, and these have consequences within an epithelial cell. The cells are tasked with expressing different proteins depending on their stress level that calls out to the immune response and says, hey, we have got something going on here. I'm quite stressed out. Come see what needs to happen. And also intracellular signals within malignant epithelial cells, like the inflammasome and pyroptosis induction, which when you have RNA and DNA in the cytoplasm or places they're not supposed to be, you have stress signals within the cells themselves, you can initiate immune responses within the epithelial cell. And then more of the theoretical side of immunology is starting to add a fourth layer, which can be called the discontinuity theory or other related theories that are being added to this, which is saying it's not just about stranger, danger, and stress, but also detecting change. So that the immune system needs there to be a deviation from homeostasis. And the idea is that a tumor can actually redefine homeostasis. So a tissue that that undergoes malignant transformation over time will start to initiate what's called a new homeostatic setting. And these set points can actually be a way of hiding from the immune response because the immune system starts to see that tumor now as non-different and it needs changes. And so things like oncolytic viruses start to take advantage of that, in which they induce a quick rate of change in difference, and the immune system can then detect that we have a change in homeostasis. And so building on all of these, the cancer cells have to be able to avoid detection from all these different layers of the immune response. And what I think we're all kind of comfortable in saying is that cancer is an evolutionary process. So this is where the evolutionary aspect of our work comes in. And this picture from Noel back in 1976, when he first stated that tumor cells undergo a series of mutations. I think we all kind of agree that these mutations then diversify and lead to heterogeneous populations of cells, which are all living within the tumor microenvironment and competing with each other. And then you will have a selection for different events like metastatic events, or when you apply a treatment, which will change the genetic and epigenetic compensation of the tumor. And in immunology, we have our own way of kind of describing this, and it's called immunoediting. Basically, immunoediting hypothesis states that the immune system can recognize tumor cells. And if the immune system is very good at recognizing the tumor cells, it can actually eradicate all the malignant cells. If the immune system is moderately good at recognizing the tumor cells, you have a lot of turnover where the immune system is killing many tumor cells, but the tumor cells are also reproducing at equal rates. And so you have this equilibrium of death and birth, which leads to a stable state within the tumor. This is where a lot of evolution happens, which can lead to what is called the escape, where you now have a lot of immune evasive phenotypes existing within the tumor, and these will escape the immune response and outgrow. So really the point of talking about the immunoediting hypothesis is that it's really Darwinian selection, that it's a competition between cells that can survive the different stresses of these different layers of the immune response. And what's really important when we talk about evolution, even in the context of immunoediting, is that we're talking about cancer cells acquiring not just a malignant phenotype, but what's considered a self-defined fitness function. So evolution doesn't occur in normal cells. Normal cells are governed by tissue signals, and once the malignant cells lose the ability to respond to these tissue controls, that is when it becomes a malignant cell that is an evolving population. So they acquire what's called a self-defined fitness function, where they're no longer listening to the signals within the cell or outside of the cell that are telling the tumor cell to they're telling the epithelial cell not to divide when it shouldn't be. And this is what makes a tumor cell the element of selection. They have a self-defined fitness function. They are no longer listening to outside systems, and they are now being selected for fitness advantages. And this is what leads us to using evolutionary biology and ecological abstractions. So ecology has really been modeling species interactions for a very long time. And so we can learn from what they've done, and we can build on it to start to understand how we can study and take advantage of the population dynamics that are occurring within the tumor and how they are interacting with the immune system themselves. And so that's really what we are looking to do in evolutionary therapy. One great example that motivates a lot of the work that I do is an ongoing clinical trial occurring at Moffitt, and now there are others occurring at other places. But this is the initial trial of adaptive therapy. Adaptive therapy looks to use ecological principles to maintain a sensitive population of tumor cells. So this trial is occurring in prostate cancer patients, and they are trying to prevent the evolution of resistance to abiraterone. And the underlying ecological concept is that you have a lot of cells that are initially sensitive. So the purple cells are sensitive to the treatment, and you can have a very small population of resistant cells. But as you apply therapy at what's considered maximum tolerated dose, so standard clinical practice, give as much drug as a patient can possibly take to have the maximum benefit of cell death. And what this does is this then kills off all of the sensitive cells and leaves only the resistant cells behind. So while initially upfront, this treatment strategy appears to be quite successful, in the end, you're left with a patient that no longer responds to therapy. So instead of applying maximum tolerated dose, adaptive therapy looks to suppress the outgrowth of the resistant phenotype, so the green cells, by leaving behind small amounts of the sensitive cells. And what this does is this allows for population competition, like you see in ecological systems, where the sensitive cells are outcompeting and suppressing the growth of the resistant cells. And so adaptive therapy gives a dose of the treatment, removes it, and while the treatment is off, the sensitive cells are allowed to regrow and suppress the growth of the sensitive cells are allowed to regrow and repress the resistant cells. And so then you apply the treatment again, and your tumor still continues to respond to the therapy. And so this on-off dosing is really the basis for the adaptive therapy trial. As you can see, there are mathematical model, which shows kind of the different theoretical ideas. So no treatment, you'll have lots of tumor growth. If you give maximum tolerated dose, you have this lovely, nice effect where the tumor shrinks below detection, will last for quite a while, but then the patient will recur and will no longer apply to therapy. Metronomic therapy also works with a high upfront dose and lowers treatment, but eventually you have escape. And this is an evolutionary consequence of continuously applying the dose without personal scheduling. While adaptive therapy looks to do these on-off doses to maintain a chronic disease that remains treatable. And so the early trial results, this update was from January of this year, shows that the patients on the adaptive therapy trial are doing much better than the control, which is standard therapy. And you can see that the black line for each patient is when they're off, and when it's red is when they're on therapy. So each patient's on-off is different depending on the dynamics of their tumor. And there are actually 4 patients who were still on the trial as of this last update. And so it really seems to be a promising mechanism of extending the treatment window for patients by suppressing the outgrowth of a resistant phenotype. And so what we asked was, we said, okay, this works well with the abiraterone. What about incorporating this into immunotherapy? We know that immunotherapy resistance is a problem. And we also know that immunotherapy can be used to tackle resistance to other treatments. And so we started to look at what we call an immunological double bind. And I'll explain this a bit as I show this project. But this is based on an isogenic cell line model, where we took prostate cancer cells, and they were a prostate cancer cell line, which received MACA radiation or 60 gray fractionated radiation, and derived 2 cell lines. So these 2 cell lines have different levels of resistance to radiation. And so we wanted to use this model to say, can we then use adaptive therapy or an evolution-guided therapy to control the outgrowth of radiation resistance? And what we found is, so you take the 2 cell lines, and what we typically do is you look for a cost of resistance. And what this means is that the resistant cell will have a fitness disadvantage compared to the sensitive cell. And typically what we do when we test this in the lab is we put them into two separate dishes. We measure the growth over time. And you can see that the radiation resistant cell line has a slower growth than the radiation sensitive cell line. And so the thought is that we could do this adaptive therapy and maintain. However, when you mix the 2 cultures together, so you take a single dish and you do a 50-50 mixture of the 2 when you grow them together, we actually found that the radiation resistant cell competes with the radiation sensitive cell in a manner that we weren't really expecting. And so what this means is, if we were actually taking, say, a patient with this actual dynamics, and we were treating them with radiation and then allowing the 2 populations to compete together, by removing the radiation, we would actually select for radiation resistance. So we know that we needed to add in another treatment option. And so this is kind of explained well in our interaction model, where you have the radiation resistant cells and the sensitive cells, and the radiation is suppressing the sensitive cells, and the radiation resistant cells are suppressing the sensitive cells, and the treatments work on the 2 populations differently. And so what we asked, we said, okay, well, what can we do for a situation where the fitness of the 2 cells does not allow for an adaptive therapy like what we see in the abiraterone trial? And so another idea that's taken from ecology is called an evolutionary double bind. And the way this is usually described is the owl-snake hypothesis, and that if you have a population of field mice and you introduce an owl, the field mice will adapt to avoid owl predation by running under a rock. Therefore, the owl can no longer attack the mice. But what will happen is, if you put a snake under the rock, you're actually sending the mice under the rock and right into the snake's nest. And so the idea is that what we wanted to do is we want to pick 2 therapies that we can use together in this kind of evolutionary double bind, where you say you choose the first therapy, you choose the owl, that will decide, or that will guide the evolutionary trajectory of the cancer cells into the snake. And so that is where we apply our immunotherapy. And the reason why we were looking at NK cells as our choice for immunotherapy is that it has been shown by us and by others that when you have genotoxic stress from things like chemotherapy and radiotherapy, you actually get a change in protein expression on tumor cells. And these proteins can have different activation and inhibition effects on NK cells. And these natural killer cells can actually, once they are activated, can target and kill the tumor cell. So the idea was that we'll use radiotherapy as our owl and NK cells as our snake. So if radiotherapy changes the surface proteins on the tumor cells enough, we should be able to take advantage of that with our second line therapy. And so we took our two cell lines and we did simple flow cytometry, where we just looked and saw what the different protein expression on our cells are. And you can see that a lot of the activating and inhibiting proteins that are expressed on tumor cells are actually altered in our radiation resistant cell line compared to our control line. And so this led to the idea that NK cells are probably likely a nice double bind for these. And so we did, I apologize. We also looked at soluble factors. So these ligands can also be cleaved from the surface of the tumor cell and can go out and activate the NK cell. And so we saw not just on the surface, but actually in the media, we see some activating ligands that are increased in the radiation resistant cells. And when we add radiation therapy, this also is increased. And so what we asked is, we said, okay, does this mean that NK cells can kill? So we took our cell lines and we mixed them with an NK cell line, NKL. And what we saw was over a 24-hour period, the radiation resistant cells had a higher sensitivity to NK cell mediated killing. And we saw this also in a mixed culture of immune cells with NK supporting cytokines, which showed that when our cells are grown in separate cultures and mixed with NK cells, that these radiation sensitive cells are more the radiation resistant cells are more sensitive to NK cell mediated killing. And so this is what we consider an evolutionary double bind. It's that one population must be sensitive to the first drug and resistant to the second. And the second population needs to be resistant to the first drug, but sensitive to the second. And so if this allows you to play with the population of cells over time, so that you can apply one drug to knock out one population and a second drug to knock out the other while preserving the prior. And so this changes our interaction model now, where we add in NK cells, which have a greater effect on our radiation resistant population. And we see that the AMC, which is our control line, is more sensitive to radiation than it is to NK cells, and a radiation resistant cell line is more sensitive to NK cells than it is to radiation. And so this is a true evolutionary double bind. And we see that the NK cells can still kill the radiation sensitive cells at a higher frequency when we have mixed cultures. So in this assay, we took the two cell lines, we put them together in a 50-50 ratio, and then 24 hours later, we measured to see who was still surviving. And what we see was that NK cells will actually kill off a larger proportion of the radiation resistant cells. And so that in this mixed culture, we maintain our double bind. But we have seen that there's an interaction effect between these two cell lines. And so it was important to study different ratios, seeding ratios of these two cell lines. So we moved over to what's called the IncuSite live imaging. If any of you have used it, you can serially image the cells over time, stain the radiation sensitive cells as green and the radiation resistant cells as red. And we seeded them at different seeding frequencies at 100% sensitive, 100% resistance, and then all the ratios in between. And what we found is that when we treated these different ratios with NK cells, you can see that NK cells kill the radiation resistant population really well when they're in separate cultures. But when you have a high proportion of radiation sensitive cells, it actually reduces the amount of NK cell mediated killing. And you have the largest effect of NK cells when you have a high frequency of radiation resistant cells, and that the disparity is highest when they are cultured separately. When we treat with radiation, we see that the radiation resistant cells are maintained even when you have different ratios of different seeding ratios, and that the highest control rate really does work when you have the highest amount of radiation sensitive cells, which makes sense. And of course, we wanted to know what happens when you treat the two therapies alone. So when we're doing a double bind, a lot of questions applies to timing and when you give the therapies. And you can see that if you give radiation and NK cells at the same time, in either the AMC population, the radiation sensitive, or the radiation resistant population, the two therapies work really well together. And at overall tumor population is highly suppressed when you have the dual treatment between the two and in mixed cultures. And so what this is really saying is that we have this evolutionary double bind, and that if the patient can receive two therapies together, that is the best choice. But also that we can use the two therapies in isolation to control the outgrowth of the different populations that we need to control in an ecological context. And we can see that when you apply the different therapies, that the ratio really matters. So if you have a high ratio or high proportion of radiation-resistant cells, the NK cell therapy is really most beneficial. When you have a high proportion of radiosensitive cells, obviously the radiation therapy is most beneficial. And you can see that we have a large change in the axes here, but the two therapies that work together work really well in all ratios and work the best when you have sensitivity. So if you can apply the two drugs at the same time, in particular patients, this would be the ideal. Our assays, like I said, this is an isogenic cell line model. All of this is done in vitro, and we have limitations. And one of those main limitations is time. We can only run these assays for about a week. And so what we wanted to do then is use a mathematical model. Jeffrey Rest, who works in the integrated mathematical oncology department at Moffitt Cancer Center, developed a nice model where he was able to look at the interactions of our two cancer cell types and our two treatments. And you can see that the simulated data from his model, along with the data that we used that came out of the experiments, correlated really well. And what he found with this model is that if you use the two therapies together, so this is 7 days basically where our experimental data stops, but you can run the model out further. And what we found is that if you have a mixed ratio of the two populations, you can actually go for what's called extinction, or you can eliminate the entire tumor by combining the two therapies together. If you have sensitive cells only, you'll be able to drive extinction with individual treatment. If you have a high level of resistant cells, you will require the combination therapy to go for extinction, but you can also get there with NK cells alone. And so what we're doing with this model now is we're trying to see what kind of dosing and timing techniques we can use to take advantage of this immunological double bind that we've identified, and go forward and do some additional experiments in vivo and with other cell line models. It is nice that we are able to find this radiotherapy double bind. I think a lot of immunologists are understanding that immunotherapy can be used really well to combat drug resistance to other therapies, including chemotherapy, radiation, and targeted therapy as well. I'm going to move on to a second project that we're doing that is kind of in the same realm, where we're talking about evolutionary dynamics. This is in lung cancer. And what we're looking at is gene conservation. So this really builds on the same idea where we're talking about populations interacting with each other and competing. So just remind everyone that selection events really results in two types of selection. So as you have your heterogeneous population of cancer cells after some time of evolution and you get a selection event, you'll have negative selection where a large fraction of the tumor cells will be selected against, and some of the tumor cells will be selected for. And what we're doing in cancer biology typically now is we look at the group of surviving cells. So we study a lot of positive selection, and we say, okay, what mutations are highly prevalent in our proportion of cancer cells that we're able to measure? And these are mutations that have given a fitness benefit to the tumor, and that is why they've taken over the population. It is much more difficult to study negative selection. So negative selection is when a mutation arises within a cancer cell that actually decreases the fitness of that tumor cell. And so when you're talking about population dynamics and competition, that mutation will actually disappear from the population. And it is much more difficult to study compared to positive selection, where you can actually visually identify them. And so we've created a new model using a robust regression model to determine genes that are under negative selection. And so what we did was we took size of the gene and number of mutations in the patients. So the idea being that each gene has a certain number of nucleic acids, and each nucleic acid has a relatively equal amount of chance of being mutated. And so a large gene will have a large number of mutations just by chance alone, and a small gene will have a small number of mutations just by chance alone. And so the idea is that what we'll do is we will take the size of the gene and map it against the number of mutations that occur in our cohort of patients. And majority of the genes will fall on this line. However, some of the genes fall above the line. And what this means is that they have more mutations than expected by chance alone. And you can see that a lot of the genes that turn up in this are things like P53, EGFR, KRAS. We're talking about lung cancer here, so BRAF as well. These are known common mutations that are seen in lung cancer. But what's interesting, though, is below the line is what we're calling conserved genes. There's actually a cohort of genes that have less mutations than what is expected by size alone. And these conserved genes are ones that are likely do not have the evolutionary space to achieve a mutation. So if a mutation occurs in this gene, it will be selected against. So they're protected through natural selection. And you can see that in this gene list, you actually get a unique sort of genes where things like ERAP and HERC1, genes that are important for survival of the fitness of the tumor cell. And so what we did is we took from the TCGA database the whole exome sequencing, so paired samples of normal tissue from each patient and tumor samples. And we looked at the lung adenocarcinoma patients and divided them up into patients who have no identifiable driver mutations, mutant KRAS cancers, and mutant EGFR. And we concentrated on the mutant KRAS and the wild type cohorts because they have a large number of mutations, and we're able to get statistical numbers to be able to compare the conservation levels between these two cohorts. And the mutant EGFR has very few mutations and is an interesting cohort that has very different conservation profiles. And so what we did was we took the number of patients with mutations in each gene and compared them with gene size and calculated the number of genes that were overmutated, so highly mutated genes, and undermutated, which we called highly conserved genes. And so anything that fell two standard deviations below or over our standard line, we looked at into further. And so we found lots of conserved genes, which was interesting between the two types. So this is one standard deviation away and two standard deviations. So these highly conserved genes, there was 160 in the mutant KRAS, 248 in the wild type. Meanwhile, highly hypermutated genes, so genes that fall high above the line of mutations, we had 518 and 593. And of course, what I'm interested in is the immunological genes, so genes that are involved in interactions with the immune system. Epithelial cells are not bystanders in the immune response. They can express proteins that can interact with the immune system. They will provide antigen presentation. They can signal through cytokines and chemokines. They can also express complement inhibitors and activators. And so what we did was we looked at 8 different gene lists based on what epithelial cell function happens with the immune response. And we found that in our conserved genes, there was a high level of conservation in our wild type cohort of antigen presenting genes, which I think has been really recognized well as an evolutionary outcome of immune selection, particularly in immunotherapy. Additionally, hypermutated genes, there was lots of mutations that we found in our complement genes between the, both wild type and mutant KRAS. And one of the genes that we're really looking into further that came out onto our list, as I showed you, one of the highest conserved genes is ERAP1 and 2. It is conserved, so expression level, and this is conservation level. So anything to the left of the line is highly conserved. Anything to the right of the line is highly mutated. And you can see that ERAP1 and ERAP2 are conserved in both mutant KRAS and our wild type, and they are both highly expressed. And what ERAP1 and ERAP2 do is it's an enzyme that functions within the endoplasmic reticulum, and its role is to break down peptides within the ER prior to loading onto MHC molecules. And so ERAP is really responsible for conditioning the antigen milieu that is expressed on the surface of epithelial cells. And so in absence of ERAP, you have an alteration in what is expressed, and certain peptides are not expressed at all. And there's a lovely murine model that showed that if you knock down ERAP in mice, you actually have expression of a common peptide, an FL9 peptide, and that this will cause complete destruction of an epithelial cell because there are immune cells that recognize this peptide already within the mouse, including naive mice who have never seen an ERAP or immune mice who have never had ERAP knockdown previously. So there are CD8 T cells within the mouse that can recognize this. And so the idea is that it is likely that when ERAP is knocked down or is dysfunctioning, that it makes the cells more prevalent to immune attack. And so what we want to do with this is we want to study further what happens when we knock down ERAP, or when we have ERAP dysfunction or inhibit ERAP function, what happens in the immune context of recognition of cancer cells. Additionally, what also came up was these same ligands that you see in the previous project, this PVR access and this MIC-A proteins that can activate or inhibit NK cells. So these are also highly expressed in our lung cancer patients, and they are conserved. So you have PVR and PVRL2, which can bind to a family of inhibitors and activation on NK cells. And what this kind of illustrates is that it is likely that NK cells have a larger effect on the evolution of lung cancer than I think we're really giving it credit for. There are lots of inhibitors that are starting to look at inhibiting this axis. Because exhausted NK cells will have a higher expression of this TIGIT and CD96, which can act like it will act as a checkpoint similar to PDL1 and PD1. And so we're looking to block these axes and hopefully turn on more activation of NK cells. In conclusion, we found that immunotherapy is likely a good candidate for a second-line double-blind therapy. This is the case, obviously, in our quite simplistic isogenic cell line model, which shows that NK cells can identify radiation-resistant cells and eliminate them faster and at a higher frequency than they do with the radiation-sensitive cells. But it is likely true for lots of styles of immunotherapy, especially the innate immune system who recognizes stress, because when a tumor cell is resistant to initial therapy, it often turns on lots of stress signals, DNA repair, and a lot of this can be recognized by the immune system. And so it is good use of a second-line double-blind therapy. And we've also found that gene conservation analysis can be used in clinical patient samples to identify important genes. So what was interesting about that project, I think when we first went in to see what genes were mostly conserved, is we were expecting to see a lot of what we call classic and immune evasion strategies, downregulation of MHC, increasing of PDL1, these kind of classic ideas. And we didn't see that. We actually saw these non-intuitive genes are being conserved. And so we think this is likely because most of the prior analysis is done on gene expression or on positive selection of mutations that currently exist. And so this negative selection of genes can be quite important. And so what we plan to do is to identify evolutionary vulnerabilities in multi-drug resistance models. So there is a lot of isogenic cell models for therapy resistance, from targeted therapies to chemotherapies. And so we really want to probe into these models and see what evolutionary vulnerabilities they have, mostly in the context of becoming more sensitive to the immune response. And of course, we'd like to do some in vivo validations of our radiation and NK cell double binds, test some of the model outcomes that are ongoing in the IMO group right now. And then with the gene conservation, we want to look at the evolutionary consequences of ERAP. It's likely that ERAP is a required gene, but only in the context of when you have an active immune response. So a lot of CRISPR analysis that has been done on things like ERAP and other genes are done in a dish that don't have any immune response in there. So we're looking to add some layers of complexity to these knockdowns and see what ERAP does in the context of the immune response. And then, of course, targeting this PVR family that binds with TIGIT, so TIGIT inhibitors, CD96 inhibitors, using things like that to get at the evolutionary kind of consequences of gene conservation. And with that, I want to say thank you. There are a ton of people who were involved. The interactions of ecologists and evolutionary biologists with immunologists and Oncologist isn't always easy. This group is quite good at listening and learning new vocabulary, and we're all interacting with each other and correcting each other's understanding of the different fields. It really leads to quite a lot of fun work, and I really do think that the reductionist biology that we're also dependent on is a footstool for this more of the systems biology responses that we're looking to do as well. And it's really great that things like Trinity, St. James's, the NIH, the Physical Science and Oncology, and the Moffitt Center of Excellence for Evolutionary Therapy really understand that this work needs to be built on with a large team, and needs a lot of time to keep going further. And I think that will be it. So I'll take any questions.
So thank you, Kimberly. So that's really fantastic and really unique. And in that context, so please include your questions in the Q&A, and I will be moderating. If there are too many, I may be a little bit choosy. But starting with some, as we warm up, and thank you also for leaving time for questions. So you first, very technical. So the analysis you did in lung cancer, you focus on adenocarcinoma, squamous carcinoma, were all comers.
Or adenocarcinoma.
Okay, perfect. So that's really exciting. So in principle, there are a couple of very simple questions. So in addition to mutations, can you consider adding CNVs, so gene loss?
So there are quite a few, because particularly this model looks just at point mutations, and so genetic loss and chromosome rearrangements and things like that will be considered in our next iteration of the model.
Okay, perfect. And Nevan Krogan in our lab and a few others, so from UCSF and UCSD, so we have a joint program focusing on head and neck cancer, which is very related, tobacco related, very related to adenocarcinoma. So from the get-go, maybe we can collaborate in some issues, so explore opportunities. And so one of the issues that come also from your presentation is the need to include the use of syngeneic animal models in which you may have the complete immune system will still be there for the analysis. So in that context, of course, there are some complications in terms of knocking out genes, etc., in syngeneic models with a complete immune system, but there are new techniques to do that. So also sort of systems biology type of approaches. So are you planning to expand into different cancers? What are the opportunities for those people who are here in the audience to collaborating with you?
So the biggest hindrance with combining all of these three right now is the human immune system is not in a dish, and it is not in a mouse, and it is very complex to try and do things like this. But the ERAP, for example, when they knock it down in mice, you're talking about a completely different gene. There's only one. There's no overlap, and you're talking about a different immune profile that's occurring. And so a lot of the work that I think room for collaboration is spanning the scales with which our work is done. So when we move into things like in vivo models, there are still going to be caveats to looking at things like especially antigen expressin, right? The HLA human leukocyte antigen isn't there in a lot of murine models. You have a lot of mismatch in recognition, allogeneic responses. So I think a lot of the room is going to be a way to branch these scales from in vitro to in vivo to understanding how it's going to work in a whole organ system is the hugest gap especially for an immunologist. It gives us a lot of headaches for sure.
Perfect. So in that sense, probably having access to information of mutational burden or mutations in clinical samples from patients that underwent immune therapies, that would be what you will benefit from.
Yes, it would be fantastic to do the same analysis following failure on therapy. Absolutely, because gene conservation is is a great window into what is required to survive. So if you change the selection event, you're going to change the conservation profiles of the genes. So figuring out what genes are essential that cannot undergo evolution during therapy, particularly immunotherapy, for me, of course, is a great next step for this project. Yeah.
So for everybody, as we wait for questions in the Q&A, if anyone in the audience is in a position to have access to or knows anyone is sequencing cancers before and after immune therapies, please.
Absolutely.
Perfect. So we identify potential open collaboration. So we may be able to help with that. So not from our center. So, but that may be a possibility. The second one, in your model, and I love your model of this. I mean, I try to see the blind. Let me go back to your model in which you got in which you have the snake in
the double blind, yeah.
Double blind.
Double blind, yeah.
So in that case, for example, if you put pressure into additional pressure by using, in addition to radiation, targeted therapies, how the model will change. So for example, to give you an example, so you have BRAF mutants, few, but you have some EGFR mutants. There are many, of course, and therapies targeting EGFR, they are routinely used. How the model would be applied? So would you use also intermittent together with immune therapy? So, I mean, how the model applies to a combination of targeted and immune therapies?
So the important thing about the double blind is that you are looking at targeting the two populations separately. So when you come in with another therapy, you have to be very careful about what population it's going to target, because if you have two treatments that target the same population, or if your two treatments target both populations, you'll get a third evolution of a third evasion method that won't be targeted at all. So if you add in something like a targeted therapy, the idea is that it needs to then drive and steer the evolution so that the second therapy you add to it takes advantage of the resistance strategies that emerge. So it really would depend on resistance strategies. So if you had, say, EGFR and you had the old EGFR targeted drug and it led to the T790M mutation, then say a double blind would be something that only targets those cells with the T790M mutation. It would be something like that. And it would really depend highly on what the actual strategy is that emerges from that first resistance group. Giving the therapies all at once is typically evolutionary unwise unless you can drive to extinction, because even just a small population left behind and you will get a resistance mechanism that you cannot target.
Perfect. I will also a question from Antoine. Hi, Antoine. Great talk. I'm a strong believer in the owl and snake approach. Do you think that combining radiotherapy with immune therapies together could increase adverse effects such as autoimmune responses?
Oh, that's a good question. I think that a lot of the idea with the double blind therapy is to choose two different drugs that have very different modalities. This comes from ecology. When you're looking, when they do the opposite, they're trying to preserve a species. We're trying to make things go extinct. With the second therapy has an absolute different mechanism. It is more beneficial evolutionarily, and I would imagine toxicity levels are different when you have different strategies. Obviously, when you start using multi-treatments, you're going to have an increase in immune response, and off-target effects following radiotherapy can really be minimized using the math models to decide an offset of toxicity and efficacy of the two therapies, because it might be best to separate them by quite a bit of time, which would then allow for minimization of therapies. But that question really would need to be answered with a little more information, I think.
Yeah, so it's a big, so in melanoma, for example, so the use of the RAF inhibitors concomitant with immune checkpoint inhibitors, so they would be going to different cancer type. It has been, there are many options, and seem to be the sequencing seem to be central. Maybe that can be another area in which your model can be applied, taking advantage of the clinical outcome from the different clinical trials and exploring what make one trial successful or sort of successful and some less successful, for example. So that can be a good option. With, in this case, melanoma, of course, is a different cancer type.
Right.
I mean, it's excellent.
Yeah, I agree.
So if you, I mean, certainly for those of us that we are very focused centric in our own world, signaling pathways in our case, and immune oncology. So you are certainly most of your presentation focus on NK because it's relatively, I wouldn't say simple, but at least in case they are there, they don't need to be antigen experience. So can you bring this also concept a little bit further into T cells, given that these are the targets for most current immune therapies?
Yeah, so a lot of work is being done on combining radiotherapy with T cells. It's very attractive combination because radiotherapy induces a lot of danger-associated molecular patterns, which activate the immune response, neoantigens, release of antigens, and it can really stimulate the antigen anti-tumor immune response. What we're looking here to do, the reason why NK cells were actually chosen was because you have this evolutionary outcome of radiation resistance. So there's radiation resistance causes new antigens to be expressed, released, and created, but that isn't the actual survival benefit, isn't tied to that. So if you remove the tumor cell's ability to then express these antigens, it doesn't change their resistance or sensitivity to the radiation therapy. So NK cells were really chosen because of this evolutionary double bind. While the T cells really take advantage of an outcome, it doesn't actually have anything to do with the resistance mechanism. It's just an outcome of radiotherapy. So the combination is very useful and has lots of practicalities, but it doesn't really fit with the population dynamic study that we're trying to do. And also the reason why I like using the NK cells, particularly in this setting, is that T cells are clonal centric. They recognize one, you have one TCR that recognizes one antigen. And so immunoediting happens very quickly. Why NK cells have a wealth of activating receptors on their surface. And so they have a broader target range. And this is really important when we are considering evolutionary strategies, because it is much more difficult to get around, at least to hide from an NK cell than it is from a T cell.
Excellent. So, I mean, I've got many more questions, but probably I can do so we have a discussion one of these days, so we can organize it. But any questions from any additional questions from the audience or from QBI? Okay, hi. Any questions? Any comments? Okay. With that point, so thank you so much, Kimberly. So certainly it's very unique approach, and we will all evolve in your direction as well. And for anyone trying to identify data sets or information about mutations in after and before immune therapies, it would be wonderful to help Kimberly move this forward. Perfect.
Yes, and just send me an email. I'm looking forward to any collaborations. Thanks for having me.
Thank you.
Bye.